Hi everyone. Tonight's video is on eukaryotic chromosome structure and how it changes throughout the cell cycle. So this is a picture of what our DNA looks like folded up into a chromosome. DNA, as you know, is double-stranded and it forms this helical structure here. And what you see is that DNA wraps around proteins. So eukaryotic chromosomes are composed of DNA wrapped around proteins. And then they fold and they fold and they fold on top of each other until they reach this structure that you are most familiar with as a chromosome. These chromosomes are located in the eukaryotic nucleus, and the eukaryotic chromosomes are linear. They are not circular like they are in prokaryotic organisms. So this is actually a picture of a replicated chromosome, and that's not what chromosomes look like in the majority of the cell's life. For the most of a cell's life, this is what a chromosome looks like. This is what's called an unreplicated or uncopied chromosome. Unreplicated chromosomes have one chromatid, so this entire structure that's composed of DNA wrapped around proteins is the chromosome, and we call this one structure the chromatid. It has this slight indented period here called a centromere. So unreplicated chromosomes only have one chromatid, whereas replicated chromosomes have two chromatids. So here's another picture of a replicated chromosome, and one chromatid and a second chromatid, and they're attached at the centromere when they are replicated. So I want to take you through how the chromosome structure changes throughout the cell cycle or life cycle of a cell. So we're going to start this life cycle of the cell in a phase called G1. So this was interesting. Early scientists named this phase of the cell cycle a gap phase because when they were looking in the microscope, they didn't see anything happening. So they thought the cell must be in a gap in its cell cycle. But when in fact, this G1 or gap phase is where the cells spend the bulk of their life, and they're very metabolically active. This is when they're making proteins and lipids and ATP and transporting things all around. This is where the cell spends the majority of the time that they're alive. During this time, cells have unreplicated chromosomes. So I'm showing you the nucleus here from a cell in G1. This cell has 4 total chromosomes, 2 large chromosomes and 2 small chromosomes. Each one is an unreplicated chromosome with 1 chromatid per chromosome. Many cells actually exit the cell cycle and go into a phase called G0. Many cells just don't divide anymore in an adult. They're still alive, they're metabolically active, but they don't, and you know what, I gotta move that. Hi everyone, tonight's video is on eukaryotic chromosome structure and how it changes through the cell cycle. So here we have a picture of our DNA, and you recognize DNA as being double-stranded and in this double helix form. Chromosomes, eukaryotic chromosomes, are actually composed of DNA wrapped around proteins, and they wrap and wrap and wrap around more protein structures until they fold up into the structure that you are used to seeing as a chromosome. These chromosomes in eukaryotes are located in the nucleus, and chromosomes are linear. They're not circular like they are in some prokaryotic organisms. This picture here is actually a replicated or copied chromosome, and this isn't what chromosomes look like during the majority of the cell's life cycle. Cells actually have unreplicated chromosomes during the majority of their life cycle. Unreplicated chromosomes have only what we refer to as 1 chromatid. So this entire structure is an unreplicated chromosome, and it has what we call 1 chromatid. There's also an indented place here in every chromosome called the centromere. Let's contrast this to a replicated chromosome. So a replicated chromosome is pictured here. It's also pictured here, but it's a little clearer here. Replicated chromosomes have 2 chromatids, and they're held together at this region called the centromere, which is also visible in the unreplicated chromosome. So how does the chromosome structure change through the cell cycle? We're going to talk about a little bit about what's happening in each phase in the cell cycle and what the chromosome looks like in each phase of the cell cycle. So I'm going to start talking about the cell cycle in a phase called G1. Early microscopists, when they were looking at cells, found that cells weren't doing anything during this large time of their life, so they called it a gap phase, and they abbreviated it G. But in fact, this is a very active time for the cells. They are making proteins and lipids and ATP and transporting things in and out of the cell. This is in fact where cells spend the majority of their life. The majority of a cell's lifetime is spent in the G1 phase. During this time, cells have unreplicated chromosomes. So I'm showing you a nucleus here from a cell that has 4 chromosomes. It has 2 large chromosomes and 2 small chromosomes. Each has 1 chromatid. So in an unreplicated chromosome in G1, there is 1 chromatid per chromosome. Many cells in an adult don't divide anymore. They're still alive and they're metabolically active, but they don't divide anymore because most adults are no longer growing. They aren't dividing unless they're stimulated to re-enter the cell cycle due to an injury. Now our cells that are still dividing, so they will maintain G1 for the most of their life, but then they'll get big enough that they need to divide. They need to divide into 2 cells. And if a cell is going to divide into 2, it's going to need 2 copies of each chromosome, so that each new cell gets a copy of each chromosome. So they enter a phase called S phase, and the S stands for synthesis, where the cell makes a copy of every chromosome. So at the beginning of S phase, cells have chromosomes that are unreplicated. They have 1 chromatid per chromosome. During S phase, which is a very complicated process that we will cover later in the year, the single unreplicated chromosome is copied into a replicated chromosome. And this replicated chromosome, again, has 2 chromatids. They're attached at the centromere. We refer to these chromatids as sister chromatids. They're exact copies of each other, so really we should call them identical twin chromatids, but we call them sister chromatids. So during S phase, each chromosome will be copied to a replicated chromosome. So at the end of S phase, each chromosome is in its replicated form. It goes from an unreplicated to a replicated form during the course of this phase. Cells then enter another G phase called G2, which again, early microscopists thought nothing was happening. But this phase is when the cell is getting ready to divide. It has to make sure that now that it's copied its chromosomes, does it have enough mitochondria and Golgi and RER to divide into 2 cells. Finally, the cell enters mitosis, which is a very complicated phase that we will cover in detail soon. Mitosis is when the chromosomes divide. Each daughter cell will get 1 copy of each chromosome. So in other words, 1 chromatid becomes a daughter chromosome in the new cell. Once the cell has gone through the process of dividing up the sister chromatids to become the daughter chromosomes, it will go through a process of cytokinesis, and that is cell division. It's very important to keep the process of cytokinesis separate from mitosis. Very often, students assume that mitosis is part of the cell division, but it's actually just the division of the chromosomes. The cell division is cytokinesis. When the cell does divide, it will again regenerate 2 cells. Each one has a set of unreplicated chromosomes identical to what the original mother cell looked like. These cells would then enter G1 and follow through the cell cycle as they were growing. So how many chromosomes do eukaryotes have? Well, it depends on the species, but it's always more than 1. If we look here at something called a karyotype is a picture of the chromosomes in an organism, and it gives the number and the size and some other characteristics unique to the organism. This is a picture of a human karyotype. Humans have 46 total chromosomes, but 23 pairs. So they have 2 copies of chromosome 1, 2 copies of chromosome 2, all the way up to chromosome 22. The last pair can be an unequal pair. This has the sex chromosomes that determine what the sex of the organism is. This last pair has an X and a Y chromosome, which tells me that this is a male. If we look at a mouse karyotype, mice have 20 pairs of chromosomes, or 40 total. Again, I see for the last pair that there's an X and a Y, which tells me that this is a male mouse. Finally, here's the karyotype of a cow. Cows have 30 pairs of chromosomes, for 60 total. Again, this appears to be a male cow. So what you should know from tonight's video: you should know that chromosomes are made of double-stranded DNA wrapped around protein. You should know that eukaryotic chromosomes are linear and they're located in the nucleus. You should know the structure of a replicated and an unreplicated chromosome, and you should be able to identify a picture as either a replicated or unreplicated chromosome. You should be able to identify a chromatid and a centromere in both a replicated and unreplicated chromosome. You should know in general the phases of the cell cycle and what happens in each phase, and you should know the chromosome structure in each phase. Finally, you should know what a karyotype is and what information it tells you. So that's all for tonight.